Hi, my name is Jade Fowler,
And my name is Genevieve Lynn.
and we are both biology majors at Chaffey College. This summer, we participated in a summer research opportunity with Chaffey College under the instruction of Bridget Kelly at the UC Riverside Department of Earth and Planetary Sciences.
The title of our presentation is Living Rent-Free, the Ecological Record of Sclerovoyance and Crusting of Modern Mercenaria bivalves from the Long Island Sound. We'll be giving you a look into the research we did with these bivalves, otherwise known as clamshells, on how we accomplished it. Now, Jade is going to start us off with a summary of this project and some data that we acquired from it.
The purpose of this research was to analyze and report the taxonomy and abundance of encrusting organisms that inhabited a collection of the Mercenaria bivalves from the Long Island Sound. We found an abundant trace of organisms such as sea sponges, polychaetes, bryozoans, capitulas that once lived rent-free on the sample cells. During our research, we used the following methods to ensure accuracy of data and accessibility of results. First, we only used shells 25% to 100% complete. Second, we measured the interior and exterior surfaces of each shell using the imagery software. This gave us a precise area measurement for the shell as a whole and at various incrustation sites. Third, we identified each encruster and used Google Sheets to collect and organize the data. Fourth, the incrustation of each shell was then calculated and reported as percent area. And fifth, we also used a Wix website to further present our sample collection and information. Now I will break down some of the results of our research. After visually inspecting the shells, we found that the organisms that mainly encrusted our Mercenaria sample were sea sponges, polychaetes, bryozoans, capitulas, and barnacles. We measured the area of each organism that encrusted our shells, and we made some interesting discoveries. We found that while 77.80% of the exterior and 79.70% of the interior of the shells were non-encrusted, sponges had the highest occurrence of incrustation on both the interior and exterior of the shells at 25.33%. This was followed by polychaetes with 14.88%, capitulas with 1.67%, and bryozoans at 0.62%. While barnacles were only found on the interior of the shells and made up only about 0.01% of the incrustation in our sample. While our portion of our study has concluded, the research will continue on with Bridget Kelly in the near future. Now, Genevieve will discuss the materials and discussion portions, as well as the future direction for this project.
So moving on to this next section here we have a small collage of photos that we took in the early stages of our research in order to catalog and analyze the large collection of primarily disarticulated bivalves, as well as their substantial fragments. We have hundreds of pictures documenting each individual bivalve, but seeing as our poster is not several computer screens wide, unfortunately, we had to pick favorites in order to exemplify some of the encrusters we encountered. So here we have pointed out some of the different encrusters and their common names alongside their scientific names. So for example, if you look to the top right of the collage, you can see some crepidula, attachment scars, and many sponge borings, otherwise known as entobias. So for materials, we use a total of 349 predominantly disarticulated bivalves and substantial fragments of the bivalve Mercenaria in this study. The shells were target collected below the high tide mark during low tide. So most of the limitations we had whilst completing this research stemmed primarily from the fact that we were working remotely as opposed to working in a lab setting due to the current pandemic precautions in place. Limitations included having only a small sampling of hand-collected shells, and as well as software disadvantages/errors. So in saying all that, it goes back to the fact that there were many limitations placed on this project because we were working within an experimental project design implemented for remote research. Hence, we had no professional photography equipment or setup as we might have had normally if working in a lab. In furtherance of this, more specifically, we were trying to take measurements of 3D shells and encrusters using a 2D medium/platform, which was another issue. Consequently, this method of data collection affected the numerical data we gathered, such as measured areas and the respective percentages that were calculated. Although working remotely did have its advantages as well, such as easily shared and accessible data for all individuals involved, as well as flexible working locations. So in the future, this research will culminate in a scientific article to be submitted to an appropriate peer-reviewed scientific journal after the conclusion of this program.
We would like to thank the Chaffey College Summer Research Opportunity Program and our University of California, Riverside Department of Earth and Planetary Sciences for making this research possible. Most of all, we would like to thank our program advisor, Bridget Kelly. Without her knowledge, direction, and leadership, this study would not have been brought to light or taken the successful path that it did.
And finally, if you want to learn more about our project and see more of our favorite pictures, scan the QR code below or follow the link to see our website and get a better look at the pesky yet incredibly cool encrusters living rent-free on these poor, unsuspecting bivalves. So thank you for
Thank you for
watching.
watching.